Well, thank you for coming, everybody, for this presentation of two students' biology research. And we will have the first one by Andy, and then who will take about a half an hour, and there'll be time for questions at the end. And then we'll move on to James. And for each of them, their advisor will introduce them.
Well, thank you all for coming. It is my pleasure to introduce Andy Albert. She comes to us via Great Falls, Montana, despite being evidently a Colorado Rockies fan. It fooled me. I thought she was from Colorado. She's a biology major and a member of the bowling team. She is also a chemistry minor with a technical writing certificate. Rob, please come on. This is based on some of her field work from last summer. It was part of an NSF research project looking at zooplankton in San Francisco Bay. And specifically, the title of this is going to be Assessing the Impact of Tissue Stains on DNA Amplification in Zooplankton. All right, Andy?
So, like Dr. Linder said, my name is Andy Albert. I'm a biology student here at Waldorf. And over the summer, I work with Dr. Michelle Youngluth at the San Francisco State University Estuary and Ocean Science Center. And my project is on assessing the impact of tissue stains on DNA amplification in zooplankton. So for starters, what are plankton? These are tiny transparent organisms that are carried along by the tidal current. Some stay like this their entire lives, while others eventually grow large enough to swim against these currents. We split them up into two major categories, phytoplankton and zooplankton. Our focus is going to be on zooplankton, and these are microscopic animals such as krill and sea snails, the young of larger invertebrates, and fish. Copepods are among the most common species of zooplankton, and they're important as they're an abundant copepod in the San Francisco estuary, and important to our marine food web as they graze on phytoplankton and are prey to fish and fish larvae. Here on the left here, I have a picture of an assortment of zooplankton under a microscope. It's a very diverse type of animal, excuse me. And then here on the right, I have the marine food web, and you can visualize better how they fit into the food web. And we're actually going to dive a little deeper into it. Zooplankton are known as a keystone species. This is an organism that helps hold the system together. And without this species, the whole ecosystem would change. So for example, what would happen if we were to remove zooplankton from our ecosystem here? Well, our pelagic predators, to start, they would have less food to feed upon, so their numbers would eventually dwindle and die out. This would create a domino effect as their predators would also start dwindling out. And on the other hand, phytoplankton, since zooplankton are their main predators, they may grow out of control and form harmful algal blooms. When these algal blooms decompose, they take oxygen from the water, and this actually creates a mass dying event. All the aquatic organisms in that area will actually die because there is no oxygen for them. And we actually call this process eutrophication. So a little bit more on zooplankton. We can also separate them by size. Picoplankton would be the smallest, followed by nanoplankton, microplankton, mesoplankton, and macroplankton being our largest. And we can also separate them by development. Meroplankton, or just the larvae of fish and invertebrates. They're going to eventually grow large enough to swim against these currents. And holoplankton will remain plankton their entire lives. They will always be carried along by that current. I actually have a picture here of some meroplankton. Some of them you might recognize yourself. At the very top are some snails, and then we have clams in that middle on the left, followed by two sets of worms, and then a couple of rhizomes at the bottom and another clam. Not only are they important to the marine world, but they're also important to scientists. As each species is uniquely adapted to their environment, and this can include light, temperature, the water turbulence, and water salinity, or the salt content. This could help scientists distinguish one body of water from another. They're highly sensitive to nutrient levels, temperatures, pollution, non-nutritious foods, levels of light, and variation in this change in zooplankton concentration can indicate a subtle environmental change that we may not be able to see ourselves. Our focus of this study are copepods. These are known as the cows of the sea. Like I said before, they are the main grazers of phytoplankton, and phytoplankton are basically plant plankton. They're under the phylum Arthropoda, subphylum Crustacea, and order Copepoda. They're one of the most abundant animals on the planet, and they inhabit a huge range of waters, including subterranean caves, hypersalty environments, high altitude lakes, polar ice water, and hydrothermal vents. And there is just an example of a copepod. So how do we study them? First, we collect them these fine mesh nets. The mesh size can vary depending on what you're trying to catch for phytoplankton. So since those are pretty small, the mesh is going to be extremely small to be able to catch them. For zooplankton, they're bigger than phytoplankton, so the mesh can vary depending on what species you're going for. But not really sure what size we used, but here we have a picture of that net. At the bottom, there's a place where you can screw on a jar. One way we are able to collect them, this is personally my favorite way, is we got on a boat and we would just screw the jar on, toss it overboard, and kind of just let it drift behind as we cruised on the water. And then we would pull it up and we would have all of our samples in that jar. The other way we could do it is we attach a weight to the mesh and we just toss it over the dock, let it sit there for a little bit, the weight will pull it down, and then we use a pulley system to bring it back up. So when it comes to preservation, we usually use formalin. It's the most common method for counting identification, but it's not usually used for DNA testing as it binds to the DNA and breaks it down rapidly. 95% denatured ethanol is used for DNA studies. It is not often used for counting as there is a lack of a well-described stain that does not inhibit DNA amplification. And that is a picture of a formalin-preserved copepod that's been stained with rose spangle. That's actually one of the stains I used in my study. Unfortunately, this lack of a well-described stain is not our only challenge, as when we were trying to find a stain that we're wanting to use for ethanol-fixed organisms, not all stains are good for seeing the organism clearly. Some can have a negative reaction with the ethanol and can obscure the sample, while certain stains cannot guide the kind of skeleton of copepods. It's a lot like the exoskeleton of an insect. It's a little bit harder. It's not like normal histological tissue stains. Or histological tissues, excuse me. So my question is there a stain that can be used with ethanol-preserved plankton samples that will not inhibit the amplification of DNA? We use the species Pseudidiaptomys forbesi as the focus of this study, as they're an abundant copepod in the San Francisco estuary. And we use a set of copepods with no dye as our control. This is actually a Pseudidiaptomys copepod there. Little fun fact, that one is female. You can tell by the egg sacs that are attached to the bottom here. We did have a question on whether or not we should remove that before continuing on with our process, as there was maybe the slightest question of it being able to affect the DNA, but we decided to just go ahead and leave them on, so you might be able to distinguish between male and female in my pictures going forward. So one of the lab techniques I used quite a bit was polymerase chain reaction, or PCR. This is a lab technique that is for rapidly producing millions to billions of copies of specific segment of DNA. It happens in 3 stages. The first stage is the denaturing stage, where using high heat, we split our double-stranded DNA into single strands. And then the annealing stage, we lower the heat and we allow our DNA primers to attach to our template DNA. And then we raise the heat again for our third stage, and our new strand of DNA is created in the extension stage. These steps are repeated in cycles, and it doubles the amount of DNA copies every time. For my reactions, we were doing about 40 to 50 cycles per run, so that's quite a bit of DNA. This specifically is more of a qualitative study. There is a quantitative PCR reaction, and we'll get more into that here in a second. So the aim of my project was to apply newly designed species-specific primers to accurately measure the amount of DNA being amplified using quantitative PCR. This would measure the change of fluorescence over the repeated cycles in PCR, and it translates directly to the amount of DNA in samples being measured. So I have a couple of sample graphs here. Ideally, the sooner a sample was amplified, the more DNA was in it initially, and we compare this to our DNA quantities known as our standard, and we're able to measure the amount of DNA in our samples. Our target species first graph would be indicated by the red, and our non-target would be the blue. And we're looking for that red line to amplify and show up sooner than our blue lines. So that would be a really good example of what we're looking for. So we tested our species-specific primers for the development of quantitative PCR of pseudodiaphanous. The aim was to amplify our target species and not amplify our non-target species. And we used an agarose-gel electrophoresis to VOC this after a PCR test. And here is a couple of pictures of our runs on that. In both of these pictures, the first two bars, bands are pseudodiaphanous. We were successful in amplifying that DNA as bands are clear and bright. But unfortunately, I'm not sure if you're able to see, you can see some faded bands in the middle on both of them. Those were our non-target species, and even though it is faded, that does indicate that we amplified our non-target. So in these particular ones, we would have to optimize our PCR a little bit more in our primers and see if we can get just our target species to amplify. Unfortunately, due to some technical difficulties at the Ocean Science Center, we were unable to do any quantitative work, but we're hoping to do some further optimization and continue this in the future. So some of these stains that we worked on, like I mentioned before, we used rose bengal. According to previous studies, this has been known to hinder DNA amplification. Watanabe actually measured the success rate of PCR amplification in rose bengal copepods to be 10% compared to 80% in unstained samples. That's a really bad statistic for the success of our own study. But this was a stain we just had on hand, and we wanted to see for ourselves what would happen. This stain doesn't stain normal healthy cells. It only works on dead cells and mucus strands. And it's usually used in the lab bed sorting of zooplankton. That's why we still had it on hand. This is a calanoid copepod that's been stained with rose bengal. So it is a nice bright pink, sometimes a dark red color like that. Helps see the morphology, and then when it comes to sorting them by species, we're able to separate them easier. Otherwise, they are transparent. It makes it really hard to see. Our next stain is neutral red. This is commonly used for live dead sorting as well. It only stains the live organisms. It's patchy or absent any dead ones, so that's pretty opposite of our rose bengal stain. This was also another one we had on hand and wanted to try out. There wasn't that many, as far as we could see, wasn't a lot of literature on how it affected DNA, so we were curious to see what we would do. In this picture, we have 4 copepod species that have been dyed with neutral red. The top row was identified as dead, and the bottom row are live samples. As you can see in our live samples, it is a lot darker, and in the top row, it's pretty faded, and even in this bigger one, you can see there's a batch totally missing. And for our black sheep of our stains, we have chlorazole black. And I like to say that because this is normally used as a fungal stain. Morimoto et al. Tested chlorazole black on parasitic intestinal protozoa and found a 91.6% success rate in PCR studies. We chose this one because it had a really good success rate out of just this study. And out of all the other ones I chose, this one was the least hazardous. So, and here is a picture of some reds that have been stained with chlorazole black. And now just getting into the methods, we researched and tested these different stains to see if they would be useful for dyeing ethanol preserved plankton samples and see if they would work for subsequent use of quantitative DNA work. We did sets of 30 copepods placed in petri dishes with ethanol. We did 1 to 2 drops per stain, and we observed the samples to see how it would react with the ethanol and if it was going to sufficiently stain our copepod. Because as I said before, that kind of skeleton can make it difficult, and sometimes the stain doesn't take. We let these soak for an hour, and we transferred them to 1.5 milliliter tubes with fresh ethanol. We did 5 copepods per tube and 4 tubes per stain. Starting with rose bengal again on the very far left there, or right, excuse me. We used 1% rose bengal. We only needed 1 drop to significantly stain the copepod. It kept it staining when we transferred it to fresh ethanol, so that does help out a lot as far as the staining process goes. In the middle is 1% neutral red. We only needed 1 drop again, but when we transferred to fresh ethanol, consistent with previous studies done, it did not keep it stained at all. As if that stain is only good for staining live organisms, and these were all dead samples. And then on the end, we have 1000% chlorazole black. Our first drop of the stain did not sufficiently stain the copepod, so we did have to add a second one. And we actually found that it did have a negative reaction with the ethanol as it formed little particles that could obscure the sample. Pseudodaphnis is a bigger species of copepod, so this really wasn't an issue, but this could pose a concern for smaller species. So here I have some of our samples under a regular microscope view. It's really hard to see the stain even with this projector. The rose bangle one, there is a little bit of a tint, but you can barely see it. They all look pretty much similar to our no-dye samples. So we used dark field microscopy to make those colors pop and be able to see how well it actually stained the copepods. Like I said earlier, neutral red didn't take at all, so it looks just like our no-dye samples. And then rose bangle and florazole black look like they were sufficiently stained. And then here I have a picture of the particles that were formed in ethanol with florazole black. Pseudodaphnis is bigger, so as you can see, it's not doing much to obscure the sample, but again, this could pose an issue with smaller copepods. So because we were unable to do any quantitative studies, we just did a normal PCR followed by an aggregate scale electrophoresis, and we compared the number of bright bands versus the number of dull and or absent bands. Ranking them from best to worst, we would start with florazole black, as it appears that the bands are similar, if not better, than our no-dye replicates. Rose bangle would follow right behind. Had a couple of bright bands, but it seems like those other two, one is faded and one is peninsula. And then neutral red was the worst. You can only see really one clear band, and the rest appear to have just not shown up. So from this, we can conclude that rose bangle and neutral red did reduce the amplification of PCR, or the amplification of DNA in our PCR studies. And unfortunately, we weren't able to get to as many stains as I would have liked, so here are a couple other stains that I was hoping that we could test in the future. Borax carmine. In an older study in 1976, Nichols did a test on small copepod DNA and found that it did not affect the amplification. But in a more recent study on parasitic worm DNA, there was a significant effect. So I'd be curious to see how that would work now with the bigger species in it, that we would have more success like Nichols did. Here is a parasite that's been stained with borax carmine, and that's what gives it that red look. The other stain I wanted to test was methylbromine. Torsten and Burton, they saw no effects when they used methylbromine on their histological tissue stains. I didn't find any literature on them testing it on copepods or another small organism or even parasitic DNA. But I'm curious to see how that would work. Down in the corner there is a embryonic mouse spring that's been dyed with methylbromine. That light color would work really well for seeing some of those morphological features that would make species unique from each other and would help with the identification in the lab. Florazole black would probably not do so well in that, since it is a darker stain and it can make certain features blend in together. So that is all I have. I just want to say thank you to Dr. Michelle Youngbluth at SFSU, as well as the Kimmerer and Youngbluth lab group for helping me out throughout this whole process. Thank you to SFSU, Esri Ocean Science Center, and the NSF for funding and providing me the workspace for this. And of course, thank you to Dr. Linder for reading endless amounts of papers for me and helping me through this as well. Are there any questions?
Is a copepod big enough to see without a microscope?
Yes, some species are, but it's like the top of a pin. It's like very, very small. You can see it if you really focus on it, but you can't see any distinct features without the microscope. Yeah.
So you said epidemic would be formal.
Yeah.
And so would the quality, would the results of stain change if you change the preservative?
So formalin is commonly used for county identification. We used ethanol for our studies. So the goal was to see if that would, since formalin does bind to DNA and breaks it down, we were hoping to see if we could use ethanol to help find stains, because ethanol is usually used for our DNA studies. So we were just seeing if we could find a stain that would work well with the ethanol and still stain the copepod, which
mentioned the other things that you're hoping to do this last summer with it, but were unable to. What are your plans after graduation? Are you going to be able to come back to this project, or is someone else going to continue on?
I imagine someone else is probably going to continue on. I don't have any solid plans to go back. I would love to go back and finish this. But I don't like living in California.
Are there many dyes to choose from, or this is just almost all that you can use?
Yes, there's actually a lot more dyes out there than I thought. My PI wanted me to look for stains that were non-hazardous, because she didn't want it just sitting in her cabinets after I was gone. So we were limited in that aspect, but yeah, there's tons out there. Yes.
What species was it?
Pseudodiaptomys formosa.
What did you choose that favorite species?
So that one is very abundant in the San Francisco estuary. So we had tons of those samples on hand, and if we needed more, it was like a 2-hour trip just to go out in the water and collect more. We usually got a good amount per trip out there.
So that abundance means, so you said you collected the sample like in the net, right?
Yeah.
So in one batch, like how much sample do you collect? I want to get some idea how abundance they are.
Quite a bit. You could probably pipette like any amount onto a microscope and get like huge, like a tons of diversity in that of different plankton.
So how far do you go with the boat to
Oh,
collect the sample?
I'm sorry.
And how much sample you have to collect there in that net?
We just, from what I understand, we just go out there and we just kind of let it float for like 15 minutes or cruise alongside us. And usually we collect about a jar full. They didn't really have measurements on the jars, but it was like probably about this big.
And then they are pretty much at the surface, right? You don't need to go to the deep below.
No.
And did you just keep them in the water, or did you have any specific conditions to keep them under, like, some percentage of CO2?
Sometimes we would keep them in just that jar of water until we got to the lab and we would transfer them to ethanol. And then sometimes we would just transfer them straight to ethanol right on the boat. But eventually they'd end up in ethanol.
So with all the stuff that's in the water, how can you just like take this strainer and say, oh, I only have that? Like, how do you pick out all the other stuff?
That's a good question. Usually there's quite a bit of plankton in there. Lots of the young of invertebrates and fish are part of that, so we can distinguish those as zooplankton. As far as like detritus goes, like little branches and dirt and stuff, that's pretty easy to separate. But I didn't really have much of an issue as far as like separating it. It was more or less figuring out which ones we needed to keep and which ones we could just like, put back.
Any questions? Thank you very much, Wendy. Well, we'll take a few minutes to set up for the next speech. I want to stand up.
I was going to ask a question about
It does make you wonder if you go swimming.
I know. They think it's chiggers in Minnesota lakes, so.
Unintentionally eating blue whale.
Yeah. You have some behind
exactly. I was like, man.
Yeah.
You even did the final audit.
I was under,
Huh?
I was under.
I know.
Are we waiting for anyone to come back in? Yes, Susie's still out, so
Okay.
you got a minute to.
Oh, be shocked. Who cares? Oh my God, I'm sorry. I don't know if you, I was telling you. So I can't do that for you. Huh? What? That's called visit. That's called what? No, I just.
Okay, so, James, actually got his idea for his project working as a roofer last summer, right? Where, it came to light that, the roofs of industrial pig barns apparently don't last very long. There's a rapid corrosion, and the farmer that he talked to blamed that on the ammonia that comes out of the pig's waste pit. And I've always been interested in screens and selections where you try to pan through many, many different things and force the one thing that you're interested in to come to light. And so we read through the literature and came up with a way to select for bacteria that would uptake ammonia. James had to actually collect samples out of one of those pits to do it. And turns out ammonia is only one of about 160 different gases that that waste is producing. That is the underpinnings of the characteristic aroma of spring in Iowa. Anyway, so he had done quite a lot of work last semester, and then we learned through Jensen and Atticus's project about another interesting thing that could be done to further James's work, and he's going to bring you up to speed and then tell you about the new.
Good evening, thank you all for coming. So I did my research on a selection for ammonia metabolizing bacteria from animal waste from a commercial hog enclosure. There's several problems associated with the ammonia. Oh, we're cut off a little bit there at the top, but there's several problems associated with the ammonia accumulation in the hog rearing facilities. One of those being hog feces are discarded into enormous waste pits under a facility, and so just to hit on about how big those are, they go down 8 feet. So however big this facility is, you can just imagine 8 feet under the length and width of the facility. All of that is filled with feces underneath. So it is very thick down there. The feces accumulate in the pit until the pit is flushed. They're typically not flushed that often. They're going to be flushed probably 2 to 3 times a year, and a lot of that depends on when farmers need manure to spread in the fields. The dense microbe populations in the enclosed pits, it's anaerobic, so no oxygen down there, and that's where the ammonia is produced. Ammonia is toxic to hogs. It reduces fecundity and weight gain, and also humans, and can tremendously lower the life expectancy of the facility's roofs with all that toxic gas under the roof. So to give you a visual, this is a rather medium-sized hog rearing facility up here. You can kind of see how dense it is with hogs. And then down here you can see when these hogs defecate, it goes through this grate here and ends up down here. This one has somehow got into. And this is a rather, yeah, I know, right? I hope he didn't get sent to market, but it's kind of hard to see with the lighting in here, but this would be a lower density. So higher density would be almost like a thick muck. Like if you're out in an Iowa marsh, it'd be very thick like that. So I'm based on how low the density is, I would say that this pit has recently been flushed within the last month or two. So the bioremediation offers some possible solutions. Conditions in the hog waste pits favor the evolution of ammonia-resistant bacteria. Some of these bacteria may be potent ammonia metabolizers. Ammonia metabolizing bacteria could be beneficial to the waste pit to reduce the amount of pneumonia in the pit. So how we selected was we made serial dilutions of the waste samples in the 2 different categories. That would be high density and low density. I gathered from 2 different areas of the hog barn where there was only a few hogs over here as opposed to where it was jam-packed, like in the picture shown earlier. Serial dilutions on minimal meteor augurs containing glucose as our carbon source, semi-toxic level of ammonia as sole nitrogen source, and we're expecting these colonies to not only be ammonia tolerant but also be capable of metabolizing the ammonia. And here's an image of some colonies we obtained from a high density source on our auger plate. So here's an interesting picture here. This is one of our colonies after the auger plate was sprayed with Nestler's reagent. And Nestler's reagent is filled with ammonia. So that would be the yellow haze you see all over the auger plate. If you focus your attention here around the colony, you can see a halo, and that would be an indication that that colony is metabolizing the ammonia. So that's very exciting. We were happy to find that. Here's a quantitative analysis of ammonia metabolism. So the colonies with different morphologies were inoculated into the liquid minimal media with the same formulation as selection plates. We kept them incubated at 37 degrees Celsius, and then they were shaken for 72 hours. The growth was assessed by optical density at 600 nanometers. Ammonia levels were assessed by absorption at 415 nanometers using the Nestler's reagent. So here is our depletion efficiency and our standard curve on the high density as well as low density. We didn't use all of them because some of them were too low to use, so we took the best results out of the high density and low density. And one that I really want you to pay attention to here is the high density 6. We saw a metabolism rate of 1.6-fold, which is pretty good. And so here are some characteristics of both the high density and low density colonies and gram staining. So one thing to pay attention to here, these 2 bottom slides, is that the high density was highly motile, whereas the low density was not motile at all. This is our long read sequencing performed by Oxford Nanopore Technologies method. And so this is how we can read the genomic DNA, all of it. So what we were able to do, and this is really exciting, is we were able to send off low density 1 and high density 6 off to the lab to where they can sequence the entire genome. And we were waiting to get that back. We have not received that information yet, but that is very exciting stuff for us. So our high density 6 genome sequenced. The genome size was 4.1 million base pairs. The number of genes found was 4,181. 88.7% of genome show 95.3% homology to Bacillus subtilis. 11.3% of the genome show 91.1% homology to Planococcus donhiensis. And then so we have glutamate dehydrogenase mediated ammonia assimilation pathway. And then also we have glutamine synthetase plus glutamate synthase mediated ammonia assimilation pathway as to pathways to assimilated ammonia. The low density genome sequence. The genome size was 5.71 million base pairs. Number of genes found was 5,345. 93.5% of the genome show 90.3% homology to Wimpsia moralis, and then 9.6% of the genome show high homology to Ptysophora arborifolia. 0.08% of the genome is a plasmid with 96.3 similarity to one found in E. coli. And then these bottom two bullet points are the same as the last slide because they are both pathways, again, to assimilated ammonia. Future directions we'd like to expand on is we would like to identify denitrifying microbes or engineer denitrification into discovered ammonia assimilators. And then also we would like to discover bacteria with nitric oxide demutase activity. And so what that means is, one way, we would like to not only turn the ammonia into nitrogen, but we don't want it to go back to the ammonia. We'd like to turn the nitrogen then into nitrogen gas to where that cycle does not keep repeating onward and onward. Also, if you look down here, we'd also, another avenue would be just turning it into oxygen, making it a more oxygen-rich environment. So, special thanks to my academic advisor, Dr. Gary Combs. His advice and guidance were extremely beneficial. Couldn't have done this without him. The hog farmer, Luke Johnson, one of my fellow Waldorf classmates, for aiding me in collecting samples and providing insight on how the hog rearing facilities are operated on a day-to-day basis, as well as about the pits. Dr. Mohammed Zinaladin of the USDA for expert assistance with whole genome bioinformatics, and Waldorf University for allowing me to utilize their facilities to conduct my research. I will now welcome any questions. Yes.
So one thing I know about you, Jimmy, is you're an entrepreneur. Do you see any direction with products to help mitigating these issues that are,
Well,
I mean, for the hogs, for the humans, for not just the roofs?
you know me well. I don't have any NDAs here to sign, but yes, absolutely. That is something we have been looking into extensively, branched off from the research. I think that there is a huge gap in the market because there's currently nothing out there. I think there could be something developed and applied to the pits to, if anything, extend the life of the roof, but also the air quality. And if you get these hog farmers, so fecundity means the amount of piglets a sow can have. So if you say, hey, if we lower the ammonia levels and all this toxic gas, you're going to get more piglets and they're going to be able to gain more weight, I think they'll be all for it.
Well, their own health as well, sounds like.
Exactly. So one thing I failed to mention earlier is me being a commercial roofer, I meet contractors from all over. I actually heard a tragic story where people pay a lot of money to have their hog pits worked on, since it's such a toxic environment. And sadly, over in northwest Iowa, there was a father and son who passed away because they were working on the metal grates, 8 feet deep. The son fell in. The father went in after him, and they were both gone within about a minute. That's how toxic the pit is. Nothing really you can do once you go in. So I think, hearing stories like that, as well as how toxic it is to the environment and the roof and all the hogs, there's something that needs to be done here. Yes.
So about the toxicity of ammonia, how does that affect the weight loss? Like, is that associated with any specific hormones, or is it just like they don't eat well because of the ammonia?
So great question, and it actually won't attribute to weight loss, it'll just affect potential weight gain. So if you can imagine being in a toxic environment, ingesting all of these toxic chemicals, you're not gonna wanna eat as much, or you might feel sick. You know, pigs are very similar to we are in many ways. So if you can just imagine, if you're smelling something bad, you might not wanna continue eating as much as you don't need to. Carla, did you have a question?
I did, but, then I forgot.
Oh, okay.
Thanks for asking.
No, you're welcome. Okay, yes,
What bioinformatics tool do you use?
bioinformatics tool.
Yeah, what source do you use
For which part?
to get the data
Sequencing.
or DNA?
Oh, okay, Dr. Combs, help me out on this one. Who did we send this to? The USDA and, I know Ames, but also we sent it to a company in Oregon.
Oh, so the question is, where did the sequencing come from, or who's doing the interpretation of the data?
How did you read the sequence?
Oh, we sent, so James had to purify the genomic DNA. And then we sent samples to a company called Plasmidsaurus.
That's right.
And if you
They sound
draw,
reputable.
if you draw a dinosaur on the back of your order form,
10% discount.
they pick yours as the winner, yeah. You get a prize or something.
Limited budget here, okay?
DNA sequencing is a very advanced technology, we don't have that.
Yes.
That level of ammonia, when they spread the manure on the fields, isn't that harmful to the environment, or does it kind of dissipate in
So-
the air quickly?
They have facilities currently. There's a man who just bought a farm here on the edge of town. I was working in his barn. His name is Steve Nelson. He has a place over by Mason. And so what they have now is they will take, once they flush the pit, they'll load up all this manure and they'll take it to a plant. And then it's a very hard process of shifting through all of this feces to then kind of make it the most nitrogen-based they can. It can be improved upon greatly. That's just all we have now. So then they will take it out to the fields. But right out to the west of town here, there was a mound. I don't remember the exact street, but sometimes they just flush the pit and they leave a big mound in the middle of the field. And it was an awful smell, and it was just there. So sometimes when there's no need, but they have to flush the pit, there is no spreading. It's just simply, where's an open spot?
This treatment that they do, does it involve cooking and stuff?
I would have to look more
Do they
into
heat
that.
it up?
I would imagine so.
You know why I ask?
Why do you ask?
Do you remember what you saw in the high-density waste?
Refresh my memory.
Wiggling all over the place?
Oh, yes, highly motile. Yes.
No, I mean, not
Oh.
bacteria.
So it was actually in our high-density sample that we collected, there were several, we're not sure if they were parasites or worms that were just in the sample, in the high-density sample. So, yeah.
You've probably heard the advice to cook your pork well done, through and through.
That, yes, that would be why. And it wasn't just one or two, it is pretty slam full in there. So there's no telling if they're not doing that, how much more worms or parasites they're just reapplying out into the open there. Another way they're trying to combat the waste pit is sometimes they'll have a huge pond dug next to the hog rearing facilities. And then so it'll simply just be pushed out to just, I don't want to say, it'll just sit there basically until it decomposes, which takes a long way, but if with anything, just dilute with water. But then that's not helping the air quality around there at all. It's not healthy for the environment. So they're getting it out from under the roof, but they're not doing anybody any favors as far as environmental standpoints, having that. Yes?
So does the environment, when they do spread it on the field, is there a requirement that they have to take it off to a place to cook it?
That is a good question, and I'll have to ask the senator the next time he is at Waldorf. Yeah,
That's what I'm guessing no as well.
right.
Yeah.
Well, let me develop something we can put in the pit first, and then we'll push for, there'll be laws. Yes.
This is telling my age, but back in the late '80s was a film called Beyond Thunderdome. And one of the things about this film was they actually did electricity for a city based on, I think, the methane off of pig feces.
Mhm.
Is this a possible thing, or what, would it be possible to somehow change the gases and get the methane, which I believe is mostly carbon and one hydrogen, I think it's like, it's sort of like a pyramid shape, if I remember right, like chemistry like 40 years ago. But is it possible to have bacteria that will actually convert what's smelly ammonia into something that would be a gas, but you can use as a biogas?
Well, what we're trying to do here is convert it into nitrogen, and as far as methane, I'm sure you could reverse engineer anything. I'm not 100% sure on how that process would work, but I'm willing to bet there is a possibility. And believe me, I don't know if you've ever been in a hog facility, but the smell, it smells like you could definitely put something in your car and it would run.
Right.
It's that potent in there.
Oh, yeah.
And they suggest you don't be in there for too long, because then you can start feeling lightheaded, nauseous,
Oh, yeah.
other such things, especially if the pit hasn't been flushed for a long time.
Oh, sure.
So I think that's a great idea, and that's plausible to look into.
So, yeah, as the entrepreneur that you are.
Right. I'll have to write that down.
Methane is one of the 5 most prevalent gases in the waste. I know that capturing is done, I just don't know how.
With some chemistry background, the ammonia can be, the best way to recycle or reuse ammonia is to make the ammonia fertilizers.
Right.
You cannot really convert ammonia to methane, because one is nitrogen compound and another is carbon compound, but ammonia fertilizers, or if you could really call it that as the ammonia gas itself, could be the way, but methane gas could be used for other fuels, biofuel.
You could maybe separate the two gases and use.
Yeah, you can definitely separate if you have that, definitely, yes, you can.
Yes. Yes.
Last question. It goes back to my botany class that I learned. Ammonia is very toxic, but plants, they are like our natural helpers who helps to reduce the toxic ammonia from the air. So did you come across the study, or did you come across the source that says like the environment, which is like a farm with pigs and with a lot of like plants covered, and the environment with less plants or with no plants?
You know, I think that would be a great research question. No, we did not look into that, but I knew where you were going with it as soon as you started to ask the question. And I personally think that would be great for further research, just to see if you could show, okay, we have more plants, and maybe which kind of plants, like the snake plant that emits more oxygen than others. If you were to place those throughout the hog rearing facility or every so often, would that lower the ammonia levels? That's a great platform for further research. I do not have an answer to that at this time, though. Okay.
I would just like to thank the department for serving subways prior to this presentation.
Yeah. Well, hopefully I didn't put you to sleep. Well, thank you all very much for coming.